Myself Mohsin Khan, a PhD scholar, Department of Pharmaceutical Chemistry, FPH RUAS. In this session, I am going to talk about pharmacophore modeling with theory and demo. In this session, I want to discuss the introduction about pharmacophore modeling. What are the different components we can use in pharmacophore modeling? Filters for drug-like properties, applications, and different types of pharmacophore modeling. Again, how to do that structure-based pharmacophore modeling. What are the open software is there with the demo. First, you have to know that what is pharmacophore modeling. The pharmacophore model is a computational technique or tools that used to identify the 3D features of a molecules or a group of molecules that for binding to a specific like receptors, enzymes or proteins. Like it is used to identify and represent the spatial arrangements. Means in that molecule some arrangement is there, like it's having that aromatic structures, okay, some hydrogen bond donor, acceptor, okay, on hydrophobic regions that helps to for that responsible for the biological activity. If all the spatial arrangement is there, then it can bind to a specific target and it show that biological effect. Next, it guides the virtual screening like lead optimization of a design of the novel compounds with improved binding affinity. Okay, when we take it structures, after doing that pharmacophore modeling, they'll give the number of structures with that similar, like a similar structures that can bind to a specific receptor. Next, from that we can the large LCM, we call it LCM, like a large compound database. From that large compound database, we identify that few structures, okay, the topmost structures that can bind to a specific structures that we call as a hits or lead compounds. Next, what are the features in pharmacophore modeling? Like hydrogen bond acceptors, it can accept the hydrogen bonds. Example like hydroxyl group or amine groups. Like hydrogen bond donor, the atoms that can donate that hydrogen bond. Example like oxygen in carbonyl group or nitrogen in amines group. What is the hydrophobic region? What the means of hydrophobic regions? Area that interact with that non-polar environments. Next, the aromatic rings. That every molecule having the aromatic rings. It can be one or two aromatic rings. Okay. Next is the charge center. Like positive and negative ions that interact with the charge region of that receptors. Next, I took an example. Like you already know that how to download the protein structures. Okay, here I took one VGFR2 like based cancer protein. Okay, in that form that structure I took an co-crystal. It is the co-crystal of VGFR2 receptors. What are the features in that co-crystal? So like hydroxyl group present in methanol group. Then which acts as a hydrogen bond donor. Next, oxygen atom in that hydroxyl group or ether and oxazole which acts as a hydrogen bond acceptor. Like it is having the two aromatic rings. Next, that methyl group on oxazole. Right? In oxazole ring on methyl group is there, it acts as a hydrophobic region. And that flexible linker that we need linker, but to connect the two aromatic ring, like we need our ether linker or methylene, like CH2 linker, to connect the or to bind that both aromatic ring. Here, like filters for drug-like properties. Okay, what other filters we can add in pharmacomodeling that enhance that biological affinities? Okay, towards that protein, it will give that highest binding affinity or it will give the biological activity. Like Lipinski rule of 5. You already all know that about Lipinski rule of 5. What is that? The molecular weight is less than 500 Dalton, and the lipophilicity log P less than 5. Hydrogen bond donor is less than 5 and acceptor less than 10. Like rotatable bonds is also less than equal to 10 or polar surface area on 40 angstrom. Next, the filters. What kind of filters we can add in that when are doing the pharmacophore modeling? Like spatial constant. What is the spatial constant? In that molecule, they are having some features is like any group functional group is there. That functional group Okay, it will be on the right portion. Okay, I having this on the aromatic ring, phenyl ring is there. If I put on the second position, okay, chlorine I will put. The chlorine is not in the second position, it will not bind to that correct pocket of that receptors. Like that is the called spatial constant. All that functional group, all in the aromatic that molecules in that correct position. Next, feature tolerance. So allow the all small derivatives in features that accommodate the flexibility. If the flexibility is there, that can go and bind to that specific target or protein. So what are the applications? In that application is a drug design. The application is there screening database of the potential hit. Already I told that from the large compound database, we have to choose that specific, like they give hits. Okay, it is a time consuming, cost effective. In the early stage of development, we can find out the hits. Next, lived optimizations. Like in that, what is by guiding the modification of the drug candidate to enhance their binding affinity, selectivity, and pharmacokinetics. Like here in this stage, what we do, we're minimizing that side effect and toxicity and enhance or improve that their features, pharmacomodeling features, which helps in that binding to a specific target. Next, toxicity prediction, like identifying the off-target interaction. In the morning session that Chad told, if I target the drug in the heart, but it will bind to the head. Okay, that you have to predict in the early stage. Okay, that if I predict in the early stage, that helps in that. Okay, developing the further, like by identifying the unwanted binding and through other receptors in helps with accessing the potential toxicity. If it is go bind to the other receptor, it may cause toxicity. That's why we have to predict that off-targeted prediction or to identify the off-targeted. Next method. Here are two types of methods there. One is ligand-based pharmacomodeling. One is the structure-based pharmacomodeling. The ligand-based means we have the only active ligands is there. In that through we have to made one like protein, like by using the homology modeling, we have to made a protein, but incorporate. Next, the structure base. In the structure base, that protein I have already on protein is there, with refined protein is there. Through in that protein, I have to make the molecules or structures that can bind or fit into that receptor. This is called the structure based receptor. Here I'm going to talk about that only structure based pharmacomodeling. So what are the requirements for the structure based we need on protein? Like the 3D arrangement, the Pharmacophore model is constructed by placing these features in 3D space that represent binding the orientation for the ligand at the receptor site. What are the steps involved in that for structure based pharmacomodeling? Like obtain a receptor structures. First you have to obtain a 3D receptor structures. Next, identify the key features. Already told what are the key features, like hydrogen bond donor, hydrogen bond acceptor, lipophilicity, aromatic ring. You have to identify and create a pharmacophore model. You have to create a model. Next, generate the pharmacophore. Create a 3D pharmacophore model based on this. Identify features. What features you want that you identified, and you create a model. Next, validate the model. Test and model is a set known ligands. Like non-ligands are there, ligands you have to validate by using that ADMT studies or molecular docking studies. Next, what are the open softwares is used there? Pharma Mapper. Next, Autodoc Vina with Pharmit. Next, Pharmagist and Zinc Pharma. These all are open softwares. Next, demo I'll show. Demo I saw want to show in that open the Pharmit. Directly you go to that Google or Chrome, search the Pharmit. In the first link you have to click that Pharmit. I already explained about that VEGFR2 based cancer receptors. One receptor I took on protein like 3VHK. Directly put the only protein that ID, PDB ID have to put 4-digit PDB ID have to put. See, automatically it came that BPK. This is a co-crystal of this protein, BPK. Next step is just submit it. You can see that the receptor structure in that protein is on the middle of this receptor. Okay, and some of the ligands, if we are when we are doing the docking, we see some papers is there. They are doing the docking, but here their ligands is the outside in that protein. But in that the co-crystal of this protein, that ligands is inside in the proteins. See that here that automated the pharmacophore model came. What are the features is there in that co-crystal? Like aromatic ring is there, hydrogen bond donor is there, and hydrogen bond acceptor and hydrophobic region. Down you can see that heat screening. See here you can add what are the range you want. Like molecular weight is there, rotatable bonds, log P, polar surface area, aromatics. How many aromatic rings you want that you can put here, and the hydrogen bond donor and hydrogen bond acceptor. Here I putting 400. Here I want up to 400 molecular weight. Like rotatable bond 5, log P can put 4, total surface area 140. It should be less than 140 Armstrong. Aromatic ring, I want 3 aromatic ring. Like next hydrogen bond acceptors to put 8. Next donor 3. After putting all the things here, you have to that such mole pot. Here you can see that Campbell database is there, ChemDive is there, ChemSpace, and last one is Zinc database. If I choose the Zinc database, after choosing the Zinc database, I have to go to next to save the sessions. I have to save the session. Next, like search Zinc. Here showing 3,326 zinc structures that may bind to that BGFR2 receptors. That may bind. Okay, I put only one co-crystal, but they are showing that similar structure of these co-crystals, like 3,326 structure, may bind to that VGFR2 receptors. Here I can minimize. I want only below 100 or below 200. I can minimize like maximum mean root mean square deviation. It is nearby one. It is good. Here put 1. Maximum score. Like here you can see that score is there. I will put 10. Next apply. Okay, filter form 3,326 are showing only 1,005. When I minimize by keeping that RMSD value and scoring value. Okay, from 3,000 to 100 it came. Okay, this 100 compound I can download it like export data is there, or you can save. It will give the data like PDB format, that full cluster they will give. You can do the docking. Again, you can see that more binding affinity, that all 100 compound do the ADMET studies. After that, you have to choose from that 100. You have to choose 20, 25, 30. That 32 you have to choose for that 10. That you have to do for molecular dynamics. It will give you the better results. Yes, here you can save that. It will give the PDB format, the full cluster, 105 full cluster they will give that compound. You have to validate by using that ADMAT, by using the PKCSM or Swiss ADME, or you can do the docking. Thank you. That's all from my side.
That I saw. But how did you put three aromatic rings? That is automated. Down you put no three aromatic rings. Correct. No, but can you just go down? Correct. Can you go down? But I think aromatic ring you put three, no? The crow crystallized ligand has to. You need. Okay. No. So how do you select whether you need how much? If somebody wants to make a new compound, how do they select? How many rings they require?
Here co-crystals having the two aromatic rings. Okay, here I choose 3. I can choose the 2 also. It will give the 2 structure. That means here only give the similar structure that can bind to the protein.
Correct.